Okay, so let's get started. Today will be the fourth instruction. Uh, this is the penultimate instruction, and we'll be having another instruction later in the day today. So, what we had discussed yesterday is about two major things. One is what are the recurring patterns in biological networks, where we had identified certain modules which we call network motifs. I think you can now identify. So these are double positive, these are double negative, and these are widely repeated across different cell lines, different biological processes, regulatory mechanisms. And we had also looked at some of these modules individually and like got into a basic introduction to introductory idea of how do these motifs manifest in terms of your physiological expression, in terms of signal response. So we had taken a look at this Goldwater quotient function, which gives you a sigmoidal output. If your experiment is showing you an irreversible switch or a toggle switch, then you know that somewhere a double positive or a double negative feedback might be operating. We also discussed some oscillatory phenomena where we had talked about time delay and time scale balancing. Okay. So now we have been armed with a basic understanding that what are the sort of components which we can expect in the large scale networks which we are looking at here. Suppose in this immune system, what are the major components or the building blocks of these biological regulatory networks? If you want to do an exercise, can you identify how many network motifs are there in this particular network type? Okay, so let's go one by one. What are the activation motifs? I'll do one for example. So this is an activation. This is one activation. Okay. What are the inhibitions? This is one inhibition. Okay. This is one. Also, there is one more. What other motives do you see? See, there is one double positive motif here. There is a double negative here, which is also essentially a positive. Okay. There is also a double negative, essentially positive again. This is a negative. So we have one double positive, two double negative, one normal. So if I now ask you that, can you identify these sort of small components from large networks? You'll be able to get an answer. Okay. So apart from the ones we had discussed yesterday, I want to discuss one more small thing, which are three component modes. So till now, what we have discussed is a double positive between two species or a double negative between two species or oscillations where you have time delay between species. You can also have three component motifs where you have certain X, Y, Z components and how they interact with each other. So they make up something called as feed forward loops, and they can be of two types. You can either have coherent feed forward loops. So why is it called coherent feed forward loops? Look at this one. See, X increases Y and Y increases Z and X also increases Z. So X through both pathways coherently increases. So the second example, if you take a look, X inhibits Y and Y activates Z, but X inhibits. So in both the pathways, X is inhibiting. So these are in the rest also you can try this out. So see, there is a coherence. Whatever is the effect through one path, the same effect is through the other path involving intermediates. And the other type is called an incoherent feed forward loop. So here you see X activates Y, but Y inhibits Z. However, X activates Z through the other path. So X is activating Z in one mechanism, but through Y it is inhibiting Z. So there is an incoherence between the effect of X on Z. Similarly, you can have this, but X is inhibiting Y, Y inhibits Z, and X inhibits. So these are a few combinations possible between three component motifs, and these are called feed forward loops because the information from X is being fed forward to Z. That's why it's called feed forward loop, and you can either have in the same sign or the opposite sign. So it's either coherent or incoherent. And these have certain biological sort of outputs as expected, since like double positive had toggle switch or irreversible double negative. These also have certain response signal curves, but we are not discussing that because for these do exist in biological system, they are quite ubiquitous. But I think if you want to take a look at it, you can take a look at it, but we are not going to further details because we have already established the most common ones. So these are three. Remember, we had talked about how does the circuitry inside the mammalian cell look. That's what we had started with before beginning the discussion on these network models. So now we have identified what the components are and how these components give individual responses. So when they are connected, they will give a collective response in terms of your physiology. And we had seen that the circuitry inside a cell should look something like this, where you have different components and they interact with each other to give a final, like a signal. Then through all these circuitry it will be processed and you will get a output for the system. So now we have identified how this signal to a cell, how is it processed. Now let us take a look at one particular process and really discuss this in detail. And that is regulation of cell cycle. So just some brief background. As I mentioned yesterday as well, cell cycle occurs in a cyclic manner through sequential phases. So initially, it's not exactly shown here, but you can have a G0 state, which is a resting state, which is outside the cell cycle, but the cell is not cyclic. From G0, when it receives certain signals, certain growth factors, or it becomes primed for cell division, it enters G1, where you have cell growth. Then it goes to G1, G1, G1. Then there is this checkpoint which I mentioned. So it has to cross this checkpoint. Then it will enter S phase, where your DNA will get replicated. Then it will move on to G2. Then it will mitotic phase, where it will undergo division. Now there are certain checkpoints at almost every one of these steps. Some are not as rigid as others. So we'll be discussing the two major ones which can actually help in controlling this cell cycle regulation process. Okay. So we'll take a look at how this process of investigating what are the key regulators of cell cycle and how do they regulate the process of cell cycle and cell division has been developed. And this will give you a sort of timeline view of how systems biology kept evolving across the years. So the very first sort of model which was proposed for cell cycle came from John Tyson, who worked in Virginia Tech, and this was published in PNAS in 1991. What is this paper is titled Modeling Cell Cycle, Division Cycle, CDC2 and Cycling Interaction. So this is the particular model. Let me explain the model in a brief. So for example, whatever you see as P here is phosphate. Okay. So for example, you have this CDC2 molecule. When it becomes phosphorylated, you have this CDC2P. Once this CDC2 becomes phosphorylated, now it can bind with something called as cyclin. And as you might guess from the name, since it's named cyclin, these cyclin proteins actually go up and down in a cyclic manner. And the individual activation and the subsequent degradation of these proteins in a sequential manner actually controls what phase your cell cycle will be. So in G1, there is one particular cyclin which will go up, then it will come down and the cell will cross to S. In S, one cyclin will come up. Once it goes down, it will go to G2, and so there is a sequential activation. And since after the division, it has to restart again, these proteins work in a cyclic manner. So therefore, cycling. And they need these sort of CDC partners to work. Okay. So this phosphorylated form of CDC2 binds with cyclic. So you have this particular. Okay. And then what you have is one phosphate is clean. And this, where you have the phosphate on the cyclin and the CDK2 is bound to it, is the active MPF. MPF is called the metaphase promoting factor. So whenever this particular active MPF is formed, sorry, that will signal the cell to undergo mitosis. So the cell will live. So this is one of the most earliest and most basic models. You see there are only two components and there are certain phosphatase and dephosphatase. So it's a most rudimentary. When the model equations were written down for this, you see, so these species have been written in terms of what we have been discussing. So differential equations. You see, most of the terms used here are linear terms. There is just one non-linear term, which is this f of n and f of n. So f of n is this certain non-linear function. So you see there's a to the power 2 here. So it's a non-linear function. It's not linear anymore. And what this essentially represents is if you follow this dotted line, see this active MPF is providing a positive signal for its own activation, right? So this what you see at the double phosphorylated form is the inactive MPF, right? Because I mentioned that once this phosphate is cleaned, it becomes so inactive to active happens, and this active also provides a positive feedback for its own formation. So more the active MPF is formed, it will start producing more of this. Right? So you have this non-linear term. Because we had seen yesterday that whenever you have these auto positive, double positive, usually we can model them by some non-linear. These were certain parameter sets used. Some were from experiments and others were adjusted accordingly to get the picture. And now they did a two parameter bifurcation like the ones we had discussed yesterday in terms of the oscillations and the bistability which I had shown, if you remember. So what they did was they did this K4 prime versus the K6. And what they observed is there are three regions. So in region A, the system is stable. In region C, the system is stable. And in region B, oscillate. Just like yesterday, how we had seen that depending on the parameter regime, you will either have a stable solution or oscillatory solution. And so from their model, after because they had adjusted the parameters suitably, what they saw is you see the time period of this oscillations is how much roughly if you say from 1640 16 to 50. So let's say like close to 30 to 40 minutes. And this agreed well with experiments because for this particular system, which is the early frog embryo, so you extract the embryo in the early stage from the frog, and in those cases, the cell cycle duration is roughly 30 to 40 minutes. So that model. However, the experimentalists did not believe this because these, if you realize, how is every portion of these equations, or how are all these equations linear? Okay? Because we have been mentioning so much non-linearity, right? So, and although in such early days, no one actually believed in this sort of mathematical modeling in case of biology. Tyson was one of the very first people, rather the founding fathers of mathematical models in biology. The experimental, even though guys who had discovered these cyclins and these proteins, even they did not believe his model. They said, okay, you might have gotten the time period, but we don't believe that the model works. You cannot use such mathematics to explain such a complex biological system. But Tyson had faith in his model. He combined with someone like Belanova. So Belanova had come to Virginia Tech to work here and they became very good friends. And they sort of had faith in this model and they enhanced this model. So what they did is, see, if you see this portion right here, so this is the CDC2, which I had mentioned earlier, and this is your cycle. And why this is a more robust model? You see, this is the title of the paper. This is a comprehensive model of M-phase regulation in Xenopus oosa. The earlier case, it was only the early embryos. But this, so Xenopus is something, it's a frog. Right? Okay, it's a frog which originates in Africa. So you can, what the you can take the oocyte, which is the ovum of the frog, and you can extract it, and then you can culture the cells for this. But this is a theoretical model. But since that was only restricted to the early embryo, but this is a comprehensive model of a proper, normal, mature cell. So let us take it. So if you see this module, the essential interaction is still the same. So what is Y and T here? Y is tyrosine, T is 3. So these are amino acids which have hydroxyl residues, so they can be T is 3. Y is tyrosine and T is 3. So these have OH groups in the side chain, so they can be phosphorylated. Okay. And you see the same sort of thing happens. So this cyclin, this binds with the CDC2 sort of cyclin partner, then it gets phosphorylated. So if you see here in the positive direction, something is written which is called V1. If it is not visible very properly, it is V1. In this direction, it is written CDC20. Let me give you one terminology. So whatever adds the phosphatase or adds the phosphate group is called the kinase enzyme, and whatever removes the phosphate group is called the phosphatase enzyme. So here, what is V1? It's a kinase. And what is CDC25? It's a phosphate. So you have certain combinations of these kinases and phosphatase, and you see there is a phosphorylation which happens, then there is a dephosphorylation by V1, then CAC is another sort of kinase which provides one more phosphorylation, then again you have CDC25 which is a phosphatase. So ultimately once you generate this one, so this is again the active metaphase promoting factor. So once this is formed, you have mitosis and cell division signal. Okay. So this is essentially the same model which was given earlier. If you take a look at the second one, we can identify some of the modules which we had discussed. Take a look, for example. This is the pre-MPF and this is the active MPF. You see, this active MPF is helping in the phosphorylation of CDC25. And CDC25, its phosphorylated form, helps in the conversion of this pre-MPF to active MPF. So, isn't this a double positive feedback? Because active MPF enhances CDC25 phosphate form. CDC25 in the phosphorylated form helps me. So there is a active MPF to double positive to CDC25. Okay. If you take a look at this model, see the phosphorylated active MPF form helps in the conversion of something which is called IE into its phosphorylated. And what the phosphorylated form does of IE is brings in something which degrades the activity. So what sort of a regulation? This is a negative. So you're armed with a lot of things here. See, earlier you have seen lot of double positive feedbacks, double negative feedback. You have V1 double positive, CDC25 double positive here with the active MPF. You also have negative feedback. So you're well primed to see oscillatory behavior, right? Because you have negative feedbacks, you have good strong positive feedbacks, right? Everything. And what they had also. If you take a look at this curve, which is the plot of active MPF concentration at steady state versus the total amount of this cycling. What they observed is this sort of curve. And from yesterday, we had learned how to interpret this. So when you have 0.1 level of cycling, you will see only one steady state, which is the low steady state of activity. If you go for intermediate regions, you will have three steady states: stable, unstable, stable. And if you further go for more higher cyclic concentrations, you will have only one stable state. This is again your bistable. And they also check that how is the cyclic threshold for this sort of. So this is, if you say the bistable threshold, how is this particular thing modulated? So you have this INH1 is one such factor. You have this TDC25. So they check some of the factors. So for example, if they increase this INH1 or unreplicated DNA, this threshold actually moves up. If you have CDC25, if you decrease this, it actually goes down. Okay. And they also did what you see is the total cyclic and active air MPA parameter, or rather, you can call it the phase point. Right? And see, if you look at the vector fields here, which have been marked, you see these are oscillatory. See. And if you want to have an idea of the time period, we have mentioned it here. If you start from 0 here, after 10 minutes you will be here, after 20 minutes you will be here. So this is the stage, this denotes the stage of maturation of the user. So at 0 time point you will be here. As you increase in terms of 10 time steps, ultimately you will reach a cyclic manner, and after 18 minutes you will complete the cell. And then again it will restart for you now. So this is a more robust one, right? But still, where is the experimental validation? Experimental validation came, so this paper, if you see, came out in 1993. Okay? Now, interesting. So there's no experiment till now. In till this time, but no experiment is. They have this proposed model, and experimentalists have rejected it. They say no, it's not possible. We cannot see this. In 2003, Tyson managed to collaborate with a group, and they published a paper in PNS in February. So 10 years later. And now take a look at the experiment here. So what your model says is you should have a bistable behavior, right? Because you have double positive and double negative. So, nice. So if you remember this bistable ratio, they are saying you should have a bistable behavior. So this active CDC2 versus total cycle should give a bistable. In certain cases, so from the model they had said that in certain cases you will have monostable when you don't have sufficient non-linearity and stuff like that, which we had discussed. But you should be able to see a hysteresis if you do the experiment properly. Now, how they did the experiment? Look at this case. So what they did is they took again this Xenopus lavis egg extract. What they did is they added something called cyclohexamide, which is a reagent which stops the protein machinery in cells. So essentially, at the moment you add cyclohexamide, whatever the protein levels were, everything will be frozen in the cell at that time. Okay. And so you essentially fix your cell at one particular starting point. So you have a fixed time point, a fixed starting condition for each of this. And what they did, they wanted to check the effect of this total cyclin wave to observe whether there's such a bistable thing is happening. So what they did is they added something called delta cyclin. What is this value? What is the meaning of delta? Delta means this is a special type of cyclin which they designed, which is non-degradable. Right? So internal the cyclin D, which is inherently present in them, that undergoes formation and degradation. They created a special type of cyclin D for external addition, and that was a non-degradable. Okay. Now take a look at this experiment. So they added cyclohexamide, and that was the 0 time point. When they did not add any cyclin D, at 0 minutes, you see that the nuclear envelope is intact. So the cell has not entered mitosis. And even after 90 minutes, the envelope is still present. It has still not entered mitosis. When they increase the dose, so increase the dose, I mean, again, they're fixed. This is for some other cell, they're fixed with cyclohexamide at 0 minute. Then they added 16 nanomolar of this external special type of cycling. Even in that case, after 90 minutes, the envelope was in. Same for 24, same for 32. Only when they added 40 nanomolars of this external special type of cycling, you see that the nuclear envelope has vanished, the chromosomes have segregated, so the cell has entered mitosis. So this is the low to high direction. How do you show the hysteresis? What they did is, instead of adding a zero, they allowed the cell to do its proper work for 60 minutes. And the cell, they allowed it to enter mitosis. Then they added the cyclohexane. So now you have fixed the cell in the case. And then they repeated the experiment. Now you see, after the cell is entered mitosis, they have fixed it. They have put in 40 nanomolars of this special type of cyclin B, and then they again wait for a certain time to allow steady stability. You see that the cell is still in the phase. At 32, it is still in there. At 24, it is still in there. Only when they drop down to 16, you see that the nuclear envelope has regenerated. So the cell has exited from phase and gone into the body. So from low to high, they have to give, sorry, 40 to enter phase. From high to low, they had to come down to 16 to exit from. This is 40. This is a beautiful experimental validation of histolytic behavior in terms of your M phase inhibition. Also, one of the other things that you can appreciate here is that, see, if you see this, when the jump up happens, it is much more drastic compared to the jump down. You see the amplitude, you see this is much smaller than this, right? So if you take a look at this graph, in the low to high experiment, you see, so this is nanomolars, so 0 nanomolar, 6 nanomolar, this is increasing external cyclic concentration. You see up to 30, there is always a low concentration. Only when 60 was added, you see a sudden sharp change. And this black and white is theoretical and experimental. You see in the reverse direction, which is this particular experiment, there is no sharp change as such, because you see this line is slanted. So there is a gradual decrease in the extension. So think of how good was a model it is, that it was proposed 10 years back. And after 10 years, they did the experiment and proved. Now you see how good models can have independent verifications. Now see, this paper which came out in PNS, this is 2003 February, right? And Tyson is one of the corresponding authors in this paper. So this is the collaborative. So the theoretical prediction which Tyson had made, Tyson and Nova rather had made, was experimentally verified after 10 to 12. 2003 February. 2003 March. Another group did the same experiment and got the same result. They simply had faith in the model. They didn't talk to the modelers. They didn't ask for experimental help for them. They had faith in the model. They designed the experiment on their own and found out that whatever the model said was correct. So even if you have to wait for 10 to 12 years, Within 1 month, you can have two solid experimental proofs if your model is good. And these groups are completely independent. So if your model is good enough, any good experimentalist will be able to verify whether your model is correct. You need to have faith in it. This group is from Prometheus, this is from James Ferrell group in Stanford. So if I explain the experimental brief, they did the same sort of experiment. They took a different variant of the cycling, and again you see that the envelope is from low to high, the envelope is intact, 100 level it is. For the reverse experiment, they have to go down to much lower concentrations, only at 25 in the sleep. And you see in this graph also, the going up, which they have mentioned, cycling reach here, but the coming down. So again, you have a distance. So they had to wait 10 years for their verification, but their model was so good that since people had been working on it, within 1 month, they themselves and someone else also managed to verify the experiment. That is the benefit of good modeling. It can provide good clues that what should you expect from your experiment. You cannot go on blindly doing experiments without having a preconceived notion that what I should expect. Otherwise, it will be a wasteful approach. That's how modeling helps. Okay. Then further work, one more work came from this modeling group. See, I'll mention this briefly. So we had discussed this V1, CDC25, and this negative feedback. So this is a double positive. This is the double negative, which is again a positive feedback. And this is a negative feedback. What you see here, what is written as this F, is a measure of the positive feedback strengths in some form, which we need not to worry, but higher the F, higher are these positive feedback effects you can issue. And what they did from their own, so this is not exactly Tyson's model, they developed the model independently, but overall feature is this. They said that only when you have sufficient strong positive feedback do you get this bistability and hysteresis behavior. As you keep on decreasing your positive feedback strength, you see the system moves from being a bistable to a monostable system. And what is the sort of what can I say? What is the effect of this decreasing positive feedback strength on your oscillation? If you see these graphs here, you see on decreasing your positive feedback strength, your strength of oscillation also goes down. You see, for very high, you have a good enough oscillation. So in a cyclic manner in the phase. As F is decreasing, you see that the oscillation is becoming weaker. And here it vanishes completely. And even if you see it, so see for very strong feedback, you have good oscillation. As you are decreasing it, you are moving here. Okay, and ultimately this is damped oscillation. Makes sense? If you also think in terms of a physical perspective, if you have strong positive feedbacks, only then will you have sufficient amount of this CDC2 cycling. Right? And only then is a negative feedback sufficient enough. So if you have a positive feedback strong enough, it will go up. Since it has gone up, it is giving a negative feedback to itself, only then it will come. If you don't have enough positive feedback, it won't go up to that extent. So that's why you see it's a damp oscillation. You have to allow sufficient sequestration and accumulation of your food to see this oscillation, which we had also mentioned yesterday, right? From the two parameters in terms of the oscillation. I had mentioned the half-life. If you have two short half-lives, before being transported, they are already decayed. So there is no accumulation for negative feedback. So this was the theoretical model. And how does the experiment show? When they did the experiment, when they measured the cycling concentration and the kinase activity concentration, you see exactly like the model in the phase portrait. It's also. So what is it? So these points which you see here, at every time interval, they measured the value of this cycling and one CDK and they plotted it. And they saw that in the cycling versus this kinase activity, it's a circular phase. And in the time domain, you see, there's a cycling rises up, it's down, oscillating, right? So this is with good positive feedback. But they had also said in the model that decreasing this positive feedback will get rid of your stable oscillations. How to take a look at this? What they did is, see, this CDC2 and V1 which I had showed in the earlier model, all were some sort of phosphorylation, dephosphorylation process. What they did is, they mutated the tyrosines, threonines, and all the possible phosphorylation sites in the CDC2 and the V1. So essentially, you have gotten rid of the active ingredients of CDC2 and V1. So there is no more positive feedback possible because you cannot phosphorylate. If you cannot phosphorylate, then what is the meaning of a positive feedback? And you see what they observed is oscillation goes. You see there are very minute oscillations here. So it is there. So how beautifully the synergy between what has been proposed theoretically and what has been done experimentally, this has evolved gradually in systems biology. It started with very simple mathematical models. They had to wait for, then they had moved on to more robust and complicated models. Then they had to wait 10 to 12 years for the first experiments to start coming in. And then they experimentally started to believe, okay, mathematical modeling makes sense. Because if you do the proper experiment, you will exactly regenerate what you. So I hope if anyone of you wants to do system knowledge or at least theoretically modeling, you at least have some faith. If I do a proper model, if I can do the experiment, or if someone else does the experiment in a proper manner, both the modeling and experiment, then you can generate very. Now, since we had been talking of the cell cycle process, where does this a cell cycle and cell division process? In our body, for example, there are different types of cells, right? And not every cell undergoes cell division and cell cycle in exactly same way. For example, most of you must be knowing that our brain cells, the neurons, once they are formed, they don't undergo division, right? They don't divide. So neuron cells won't. So what is. So what are these growth factors? You can assume that these growth factors are some molecules which are present in your usually the circulatory system in blood, which provide signals to your cells that okay, now you should exit from your quiescent phase. So most of our cells are in our body are usually in the G0, which is the resting phase. They do not undergo active division. Once they receive this signals from these growth factors, they take the decision whether I should enter the cell cycle or whether I should. Now, how do cells respond to this sort of growth factor signal? That is also so neuronal cells won't be. Once they are formed, this will form. Okay. Some cells have to be highly responsive. For example, stem cells or epithelial. For example, and some have to be minimally responsive. So, for example, if you have an injury, the cells around that we have to respond very fast to growth factor because it has to heal your wound. Some cells which are not needed, so once it is formed, it can perform its function. So there is a differential effect of growth factors depending on the type of cell which are working. And also, if So there has to be a strict balance, right? Because uncontrolled cell growth or cell division will give rise to tumor circuits. So the decision making has to be such that cells will grow, they will divide, because you as a human being also have to grow up, right? You are growing every day towards every day. So your cells have to grow and divide, but it has to be in a controlled manner, because uncontrolled cell division will lead to cancer. So you need to grow, but in a controlled fashion. Also, so how does this control operate? Remember yesterday, I had showed you something called the EQF switch, which I had said acts as a checkpoint at this G1S sort of transition switch. And this EQF actually, along with something called as RB, which is a retinoblastoma protein, you can call it RB, the name is not sure. EQF combined with RB controls this restriction point of your G1S checkpoint. And the combination of these two and their corresponding signal actually decides whether the cell should be unresponsive to your growth factor signal or it should be sensitive and start responding. So essentially, you see something which is here, that cell division and cellular proliferation is not exactly the same. Proliferation essentially means growth, right? Proliferation. So even if your cells grow, they might not necessarily divide, because there are distinct phases, right? There are growth phases, synthesis phases, and division. So depending on each of these decision-making regulatory checkpoint proteins, for example, like EQF and RP, your cell might grow, but it may not divide. So essentially, this is a very important point, that if you control the division of cells, you're not controlling the entire growth process. Your cells may keep growing and keep growing, but they won't divide. So we'll be discussing this RB EQF. So as I had mentioned, EQF is a transcription factor yesterday. When EQF is low, the cells are stuck in the G1. Once EQF switches to a high value, it transit into this X phase. So how did the model come? Because we're discussing. So Novak and Tyson in 2004 proposed this restriction point control of this mammalian cells. Okay. See now, it was in 1993 where we are working with early embryos, Xenopus oocytes. These are small organs. Gradually, we are moving into more and more complex organs. This is a mammalian cell. So this is a pretty big model which encompasses all sort of proteins which are known at that time in circuit. What we'll be focusing on is this particular region, which is the RBEQ restriction point. Okay. So how does this RBEQ essentially operate? So normally when EQF is present, RB remains bound to it, and the level of EQF cannot increase. However, there are certain keys which are called this cyclin BCDK1, cyclin ECDK1, which I had been mentioning earlier, the CDC2 and cyclin partner. So in mammalian counterparts also there are these cyclins and CDK partners. This is cyclin B, CDK4, cyclin not cyclin B, CDK1, but mainly these cyclin D. And what they do is they come and phosphorylate this RNA. And when this RB becomes double phosphorylated, EQF is decreased. It dissociates. And now, since EQF has become free, remember I had shown in one model that EQF can activate its own transcription. And also, you see that this EQF is increasing this cycle. So once EQF has been released, it can start giving these positive feedbacks to itself, either through the autopositive and through this cyclin E and A mediated. So Essentially, let me repeat, RB remains bound to E2F and keeps it in a low amount. These cycling CDKs come and phosphorylate this RB and it detaches from E2F. And once free E2F is present, it will manage to increase its own level. So this is what we proposed. Again, this was published 2004, right? 4 years later, a beautiful experiment was done, a glimpse of which I had shown yesterday. Let us discuss that experiment in detail. And also, since also one more thing which I just mentioned, since this is a positive feedback, again we expect some sort of a bistable behavior, a toggle switch type behavior. So they propose that bistable switching behavior will exist in this RB machine. 4 years later, the experiment was over. So what does it mean this bistable switching? So they work with a simple. So what I had mentioned earlier, see, this RB remains bound to E2F. However, this inhibition, so this inhibition is essentially the phosphorylation. So phosphorylation of RB deactivates. So you see, the cyclin E will phosphorylate RB deactivated. Cyclin D CDK1 partner will phosphorylate RB. So the cyclins and CDK partners, what they use. So I must mention why do they phosphorylate? It's in the name. CDK means cyclin dependent kinase. So because they are kinase and they are cyclin dependent, so they depend on cyclin partners and they are kinases, so they add phosphorylation. Naming makes sense, right? The nomenclature. So after the RB becomes double phosphorylated, E2F is released, and then through this autopositive, and also see to a certain extent through this also. See, once E2F becomes free, it can activate cyclin E to do more and more E2F will start being released. So there is this autopositive and there is another positive feedback here. Now the experiment which I had shown a glimpse on. How do you show bistable behavior? This is the biological setup of the experiment. What they did is, since E2F is a transcription factor which I mentioned, so what it does is, it sits at the promoter region of user. So if some of you don't know what user, so if you have a certain gene, to convert it to mRNA, you need transcription factors. And these transcription factors come and sit at certain sites, which are called promoter sites. So this is sort of the switching on recognition site. So once the transcription factor binds to this promoter site, the gene will start being transcribed. Look at what we do. This is a plasmid. This is a molecular biology. So what they did is they took the section of E2F1 promoter. So since this is the E2F1 promoter region here, E2F can come and bind here. And also from this model, you see this MYC, which is also a transcription factor for E2F. This can also come and bind. But let us focus on this. And with this E2F promoter region, they attached a green fluorescent protein. So essentially what should happen? More the amount of E2F, more will be the binding to this promoter site, more will be the transcription of this GFP protein encoding gene. So more will be the green fluorescence. Make sense? Clear to everybody? So essentially, higher the E2F, higher will be a green fluorescence. And you should also see some sort of hysterics. Right? So, how to do? So if you don't have a bistable thing, you should expect something like this, right? Where you have a monotonic. But if you have a bistable, you should see a low, you should see a high, and at some intermediate concentration, you should see. How was the experiment taken? For the low to high situation, what they did is they take a look at the red lines. Wherever you see red, so without pulse is mentioned in red, and the red lines in the. So what this is something called the FACS experiment, which is fluorescence assisted cell sorting. So it separates cells based on the fluorescence intensity. So whatever is low intensity will sort it out. Whatever is high intensity cells, it will separate. And this is a histogram. So this is essentially the number of cells at each intensity. So this is increasing GFP intensity in the right hand direction. And higher the peak, that is more number of cells are at that particular intensity. See the rate curve. 0.02% is low amount. So as they increase from 0.02 to zero 0.2 to 0.5, you see, follow the rate curve in the histogram. It is still low, low, low. When they move to 1, you see a small peak is appearing in the high region. When they further increased it, this peak has shot. What is essentially means that at low values of serum, you had only low EQF cells, which are low GFP, essentially low EQF. As you're increasing your serum percentage, your high amount of GFP cells are coming. Once they move 3%, you see there are less number of low intensity cells and more number of high intensity cells. So this is this behavior. Now, how do you do the reverse experiment? This more what they do is they put a pulse of 20% serum, which is extremely high. So once you put a 20% serum pulse, all the cells are transited to the high state. After providing the pulse, they remove the pulse and put a sustained serum percentage, and these percentages are these 3%, 2%, and so on. Now what happens? If you go in the reverse direction, so so follow the green lines. No? So you provided a 20% pulse, so all cells are now in high. Then you remove the pulse and maintain some serum percent. You see, at 3%, there is more number of high, more number of high, less number. When they decrease it to 2%, still there are more number of high, less number. 1%, 0.5%. Only when they drop down to about 0.2% or 0.02%, did the amount of low EQF or rather low GFP cell become more than the high GFP. So essentially, from the high to low direction, they had to drop down to a much lower value of serum percentage to start seeing low population cells. This is this. Makes sense from experimental perspective. So what we had shown yesterday, what we had shown today makes sense, right? You go from one direction from low to high, it has transited at a higher level. If you put a pulse and fix at the high level and then bring it down to low level, you have to go back to much lower concentrations. So this establishes that indeed there is a bistable switching mechanism for the RPE. Now, this is the 2008 verification of a 2004 model. Some extra things have been done. This particular paper is from our lab, which we have worked on, which is a bit more detailed model. As you can see, there are more number of sort of interaction molecules here or interacting species here. Just still a RB2 sort of restriction point. And this is a completely theoretical. There, from our lab, they have proposed that to be a tri-stable, but this four-stable is very close. So this should be a bifurcation picture. And theoretically, through computational modeling, they reproduce this experiment where they put this pulse 20% and then allow and you put without pulse. So you see this without pulse, this see, when you put a pulse, initially the cells will be there. But see, you have put your final growth factor percentage. So initially there is a spike, but in the long term behavior, it has still approached your lower state. And there is something here which I'll reveal in the final. So did you get an idea of how the modeling of one process has evolved across the years? You started with a very simple, almost a two-component model where you had only one cyclic dependent kinase and one cyclic. From that model, they moved to a more scale model. Still, no one believed that. Then they had the experiment for that, and gradually they developed the model more and more experiment. And that is how you progress to the modeling of a particular process in system biology, maintaining a synergy between your theory and the experiment which you want to do. Say, if I want to show you the current state of how current systems biology is done. Before that, so for example, I showed you this RP2. So after that Goldbetter termination in 2009, they published a more complicated cell cycle. So you see the complexity is rising across the years. It's more and more literature is being published, more interactions are coming into the picture. They are becoming, you have better improved models which can sort of give better ideas or reproduce your experimental phenomena better. So this is one such model, which is a very famous model. Cell cycle, right? Cool. Now, this is the current state of the art system value, and I'll give you this picture in one slide how it is. So this is the latest cell cycle model which is proposed from Harvard. This is for a cell model, which is a particular cervical cancer. So you see there are a lot of components here. You see some obviously components which have been discovered already. So this will be covered. So you can see these RB2F module. You can see I mentioned cyclin B and cyclin E. See, there was also a cyclin B which I mentioned. See, there is CDC25 and E. So you have to base whatever model you're doing, you have to base it on previous established models. You cannot do something completely out of the blue and expect people to have paper. So there are certain established models. You take the core framework of that model and you build upon that. And then you check that how good is your model. This is a theoretical model. So this is how you do the theory portion. You take whatever models are published, you try to couple them together to generate a better model or a more robust model which can capture more phenomena. This is the cell cycle. If you do this cell cycle, so since I've colored this here, so this red module controls the G1 here, and the yellow and the green module they control the SG2. So whenever these are high, so whatever in the red portion, when it becomes high, the cell transits from this G1. So RB becomes phosphorylated high in the sense that E2F becomes high, it will transit from this red to yellow module, and then the downstream signaling process will carry the cell to the SG2. I'll show you one small tool which is used in experiment, at least in current systems, which is called the Fouquet reporter system. What essentially it does is that if you see in this particular thing, you see something called as Gemini here, which I'm circling, and you have a CDT1, Gemini, yeah, sorry, Gemini and CDT. Since these two are operational in distinct phases of your cell cycle, if you can monitor their activity somehow, you can distinguish what stage your cell is currently. So to this, so and this technique follows exactly the same one which I showed earlier. So you take the E2F promoter and you attach GFP to it. What you do is you take this CDT1 and fuse it with a red fluorescent protein, which is called M-cherry. And to this Gemini, you put a green fluorescent protein, M. So when the cell is in the G1 phase, see initially in the G0, there will be no color because both. In the G1 phase, this M chain intensity will slowly start to come. In the SG2M, the green intensity will grow up. So definitely at the intermediate, you have the red going up and the green starting to come up as well. So the intermediate color will be here. So you have red in G1. At the transition point, you have yellow. Beyond that, you have green. This is how it should look. Does it actually look like this? Let me show you a video from it. So these are two symbols. The human symbol. You see, color intensity of red goes up. You see, the red is going up. Then there is a spike of yellow, and then goes to green. This is a live cell imaging cell. So you take cell, you have so this is transduced cell, right? So this Fouki reporter has been transduced into the cell in the lab with two cells. So this is for HeLa and this is for XXL, two different cells. Once you put this in, you can monitor which phase your cell is in, and you can do imaging setup, right? So you take these cells, you put them under the microscope, and you take continuous images for 24 hours. And once you have that, you can now do image analysis and extract what is the duration which is staying in each phase, how much time is the total cell cycle time, what is the division, what is the total cell cycle duration, what is the G1 time, what is the SG time. Okay. So this is how experiments are done. This is the deterministic model. This is also show that how can you from the so you will get certain predictions or outputs from this deterministic model from this response, and how do you monitor them using variable. So whatever I talked about in this course, mostly phenomenological modeling, which I had mentioned in the first. To a certain extent, you can there is some incorporation of data-driven modeling here based on these experiments. For example, suppose it's actually not the life cell imaging experiment, but experiments can be done where you find out the suppose the binding constant or the Michaelis Menten constant, and you input that data. But strictly speaking, that is not data-driven modeling. Right? The absolute state of the art modeling, which is data-driven modeling, how it operates is I'll show you one picture. Something like this. So in this picture, each dot you see is one cell, and this is something called as a single cell RNA sequencing experiment. So what it does is it takes a large population of cells, suppose say 10,000 cells, and it measures the RNA content in each cell. So you have a count of what your gene count is. So from gene name RNA. So suppose say in cell number 1, what is gene A? How many copies of gene A are present? Gene B, how many copies are? Gene C, how many copies? For the first cell, you generate. For the second cell, you have one cell. For the third cell, so you generate a very high dimensional data, right? For example, if you have 10,000 cells and 20,000 genes, you have a 10,000 cross 20,000 matrix. And then using algorithms which involve a lot of machine learning and even AI nowadays, you can bring down those data sets into your appropriate dimension. And then if you plot them using a lot of computational software, and you can actually start segregating these cells either in G1S, MG1UC, this is G2M, M, based on the genetic content of individuals. This is very powerful, right? Because whatever modeling of the experiment. So, for example, the FACS experiment which you are doing, you are getting population level picture. Right? If you do it 10,000 cells, you are getting the histogram, which is an average picture. But it might also happen that not each cell is behaving exactly in the same way. It is not possible because there is some sort of randomness, there is some sort of heterogeneity inherently in this. If you do experiment at single cell level, you can actually probe this heterogeneity. So even in a population of cells, what is your distribution of cell cycle? Not every cell will divide at the exact ratio. So this is where the state of the art single cell RNA sequencing. So that was the basic outline of how system budget developed in the context of one particular problem, which is cell cycle regulation, and how state of the art model in terms of cell cycle and other regulatory proceedings. Yeah, that's it for this class. I had kept one ticket which is for the last class will be shown in the last class, which I'll tell you. So we'll discuss something called as stochastic simulation. And why we need to do that is we need to study the effect of noise in biological systems. Noise, which is you can also call it as fluctuations in biological systems. Noise and how to simulate. And I'll show you one or two small problems of how you can do stochastic simulations of small biology. So we'll meet at 8 p.m. Okay. Thank you.